Welcome, students, to today's exciting exploration of one of the most fundamental topics in microbiology, bacterial structures, general morphology, and composition. Have you ever wondered how these tiny organisms, too small to see with the naked eye, are capable of surviving in nearly every environment, from the human body to the deepest oceans? The secret lies in their complex internal structures and unique compositions that give them both strength and adaptability. In today's lecture, we will embark on a detailed journey to uncover the key components that make up bacterial cells. From the cell wall that gives bacteria their shape, to the flagella that allow them to swim, and the nucleoid where their DNA is stored, we will understand how each part plays a crucial role in the bacteria's survival and functionality. What you will learn today: the basic structure of bacterial cells and how each part contributes to their function. Key components such as mesosomes, ribosomes, and pili, and their specific roles in bacteria's everyday survival. The relationship between bacterial morphology and their ability to cause disease or thrive in extreme environments. By the end of this lecture, you'll have a deeper understanding of how bacteria function at the cellular level and how their unique structures.
Good evening, everyone at home. My name is Adaptability Michael. I remain your lecturer in microbiology. Today, we want to look at the general body plan of a typical bacterial cell. How do they look like? If anybody is talking about a bacterium cell, how do they look like? On the screen, what you have is just a generalized structure of a dividing bacterial cell. If you look at the structure very well, you will see that this is made up of 10 different components of a bacterial cell. Time will fail us to start talking about the evolution of a bacterial cell. Don't forget that in the earlier video, some months ago, we looked at the classification of bacteria. That is, they according to Woese classification, who define living organism into three different domains of life. The first one is archaea, which is the most primitive. After that is bacteria, that is a prokarya. After that, eukarya, the most developed is eukaryotic, that is eukarya. Now, a typical bacterial cell that we see today is a replica of what they have been in the past. That is, it is telling us that now we are talking of about 10 different structure of a typical dividing bacterial cell. Then the structure was not as distinct as this. Then we didn't have a bacterial cell that will be in form of this, but as a result of evolution, and don't forget that evolution is talking about development with advancements, which depend on the environment. It depends on the molecular and genetic composition of that organism. But we have seen today that bacteria has conquered a lot of niches. You find them everywhere. That is, they are cosmopolitan. You can see them everywhere, even where life supposed not to take place. So that is talking about the evolution of a bacterial cell until what we have today. Now, this is a typical dividing bacterial cell. So on the screen, we have cell membrane. Okay. After cell membrane, we look at mesosomes. Can we see mesosome? After that, the bacterial cell also contain capsule. After that capsule, we look at ribosomes. Represented on the screen is also cell wall. Apart from that, we have what is called nucleoid. It is not nucleus. So that's, you have to take note of this. This is one of the differences between a typical prokaryotic organism and eukaryotic organism. Eukaryotic organisms do not have a specific nucleus. All organelles that you found in eukaryotic cell are no longer there. They are not specific. You cannot point to any point where you have all these organelles. So you have nucleoid. After that, you can see where we have our cytoplasm. Then we can see plasmid as well as a pili. On this other side is the flagellum. So I want you to just bear it in mind that as a microbiologist, so to say, you need to start your journey in microbiology or your journey in this course, which has to do with you understanding what microbial physiology is all about. That is, whatever function, whatever role that a bacterial cell is playing in the environment, is it as a commensal, is it as saprophytic, is it performing the function of a parasite? So whatever role that a bacterial cell or a bacteria is going to perform will definitely depend on the structure, depend on the component. It is just like in human being. When human beings do not have hand, for example, if somebody doesn't have hand, that is going to restrict his performance in to be involved in whatever is not going to be, whatever task that is not going to involve the use of hand. So the same thing, whatever you do not, whatever structure. So that is why you need to know the body plan. You need to know the composition of a typical bacterial cell before you'll be able to do, you'll be able to know exactly how do they perform that role. That is the reason why this topic is very, very crucial for you to be able to know the general morphology, that is the general appearance, the general body plan, and the composition of a typical bacterial cell. So that's going to help you to be able to know the role that they will perform from time to time. Just like I told you, a photosynthetic bacteria will definitely contain some of the components or composition that is going to make it to be able to perform a photosynthetic role. That means out of all the components, there must be a typical component that's going to start performing that role. And that's the reason why bacterial structure is very, very important. So just like I said, bacteria can exist in spherical form. You call them cocci. When they exist in spherical form, they are called cocci. They can also exist in rod shape. When they exist in rod shape, they are regarded as what? Bacillus. And when they are also curved, if the rod-like structure is also very curved, you call them fibrio, like Fibrio cholerae. Okay. Then there are some like spirulum. There are some like tetani, Clostridium tetani. They have a spiral form that is like a spring. So they have like a spiral form, and there are some that we combine the two together, cocci and bacillus, and that's why some of them also exist in the Coccobacillus. So if they exist together, if they exist together, that is in two, that is diplococcus, that is two cocci coming together, two coccus coming together, they are you call that shape diplococcus. And if they are in four, they are tetrad. And if they are now occurring in chains, that means they are streptococcus. So time is going to fail us to be able to know, to be able to go into the detail of all this. But don't forget, what we want to do today is just to introduce you for you to be able to know the components of bacterial cell. How would they perform their role? What component do they have? So it's just like I said, human being, you have your eyes. What do you use your eyes to do? If you don't have eyes, are there other things that you use instead of the eye? And that is where evolutionary development is going to come in. Don't forget that the nature has its own way. According to the theory of evolution, nature has its own way of eliminating, of less developing the organ or the parts that you are going to require less. So that is telling you that, just like I said, whatever you see in any bacteria today, whatever you see in any microorganism today, it is not just there for being there sake. It must perform a specific function. And that specific function, it is going to develop over a period of time. So don't forget the theory of use and disuse. So the organ or the parts that an organism tend to use more, that organism is going to develop more and is going to specialize in that organ or in that part. So you need to make sure that that is established here. Now I want you to take note of this structure. Can you see? Just like I said, it is not nucleus. You can see that all the parts, they are scattered. They are scattered because there is no membrane bounded. No membrane bounded. When you are talking of eukaryotic organism, that is, you are talking about a cell. A typical eukaryotic cell is going to have a membrane bounded organelle. There are some organelles that are going to be naked, and there are some that will be found within a specific confinement. So such is not the case of a typical bacterial cell. So just like you can see here, look at this, look at this, look at this. This is called the envelope. This is called the capsule. And it is not all bacteria that can form capsule. Why do they form capsule? They have their reason for doing so. So whatever you see any bacteria or any organism doing, just like I told you, that it must be there for a certain reason, to serve a certain purpose, to perform a specific role. So it is not just there for just being there for being there sake. So it has its own purpose, and you can see, apart from the apart from the pili that are numerous, there are many that are numerous. So this one also has what? It also has what is called a flagella. It has a flagella. So then I want you to pay attention to the ribosome. The ribosomes here, they are scattered. They are not localized. So, and if you look at the structure of a typical bacterial cell, there's nowhere you talk about Golgi apparatus. Nowhere you talk about endoplasmic reticulum. either rough endoplasmic reticulum or smooth endoplasmic reticulum. It is not there. So that is actually telling you that the functionality of each component of a bacterial cell will go a long way to depend on the niche or the ecological niche that microorganism is capable of doing. So if you are talking about a virulent microorganism or a virulent bacteria, that means here, in addition to all this, they are going to have a specific component that is going to be very, very much responsible for the virulence. So now, when you now see this one to be typical bacterial cell, a virulent one will now make sure that it modifies any of these parts to be able to perform that role of virulence. So that is one of the reasons why some bacteria are more virulent than the other one. For example, now, it's just like in human. Somebody who is going to be a boxer or somebody who's going to be a wrestler, that means that person should be able to develop more muscle, engage in exercise, regular exercise of bodybuilding, than somebody who is just going to be a classroom teacher. So the same thing is applicable to bacterial cell. So I want you to note, I want you to know all these components very, very well. And I am going to devote different, I'm going to devote different videos to be able to tell us the components. I will be picking them one after the other. We'll look at the component. For example, now in cell membrane, what are the other components? What are the structural components of cell membrane? How do they look like? If you see a cell membrane, what are those that is, for example, now if you see in a very big building, when you see a sitting room, what are the components of that sitting room? So the components will now depend or will now depend on the function that sitting room is going to be like. If the sitting room is too tight, that is telling you that whenever you want to host a very big meeting, you will not be thinking of making use of your sitting room. So you'll be thinking elsewhere. And I want you to be to take note of plasmid. We are going to talk about this plasmid in the long run. But at this point in time, this plasmid is what the bacteria, any bacteria that contain plasmid, that is telling you that it is a special bacteria. Because that component or that structure, bacteria normally use it for a different purpose. And those ones that have a pili, why would some bacteria have pili at the same time also have flagella? There are reasons. They are not going to have it just for a having it sake. So they are going to have it for a reason. Ribosomes. Don't forget that that is the cooking pot where proteins are being cooked. So anyone that have a ribosome scattered, the number of ribosomes that you found would depend on the functionality of that. And those ones that have mexosome, what do they use mexosome for? Not all bacteria that will have that. So without wasting our time, I want to believe that today you must have learned the general body plan of a general bacterial cell. If somebody is asking you to draw a typical bacterial cell, you should be able to know how to draw them and what are those structures that you will put. Then apart from that, we have been able to also look at what level of existence can we talk about a bacterial cell. We are talking of them existing in rod-like, they can be in spiral-like, they can be in rod-like, they can also be in sacini, they can be in fusiform. All these are part of what it is expected that you must have learned even before now. Now, I also mentioned about bacterial cell can also exist in diplococcus, that is, when two are existing together. I talk about tetrads, that is, when four are coming together, and they can also exist in chains. They can also exist in chains. They can also exist in bunch-like, that is staphylococcus. So, and I have typically mentioned specifically that plasmid are very, very important structure that you need to know. And so in this typical bacterial cell, I want you to take notes, take specific reference to plasmid. I want you to know of ribosomes. I want you to know about capsule, cell membrane, cell wall. All these are what you should be able to know. And don't forget that having known that, that will be able to tell you what are the role, the ecological role that such bacteria is going to perform, and which depend on the parts, which depend on the components of that bacteria. Don't forget to expect more videos in both Omega Bionexus and Omega Bio-Edutainment, which is your educational video on biotech related topics. So don't forget to subscribe, as well as comments. If you have any question relating to any topic, you are so free to let me know in the comment section. So then I want to see you in the next video. Thank you.